Hey Hayden, how's it going?
How are you?
What's up?
I said good. How are you?
I'm doing all right.
Good.
We've got just about everybody here. All right. A couple of announcements. Today we'll be online, and then Friday, and I suspect Monday also, we will be in person. So on Friday, show up in White Science 205. We're going to start the process of live scripting the stuff that we're going to start talking about today. So better to do that while we're all in the same room. The other thing is, in the cheat sheet section of the Moodle site, so up at the very top where the syllabus and the daily schedule and all that are, there's a section called our cheat sheets. There is a cheat sheet in there for R Markdown. And R Markdown is one of the things that we will use to turn in assignments from here on out. So you've taken the one exam that there is in this class. That's what you did on Monday. And the rest of the assignments in the class are essentially homework assignments designed to get you to work through the examples either by hand or in Excel spreadsheets to get you used to how the method that we're looking at that time actually functions. And then it's good practice that as you're working that out by hand, that you also run that in R so that you can see, first of all, check to see if your homework is correct using R, and the other thing, just to practice scripting in R. And then the second thing that you'll do for each method that we take in turn is you will get a larger, more complicated data set, and a data set that really is best worked out via R. And you will basically write a report on that. And so in both cases, what you'll turn into me are two products essentially. One is a written summary of your analysis, and I'll show you an example of that after we've kind of worked through the single factor analysis and variance. And then you'll also present to me an R Markdown script. And the advantage of R Markdown is it shows the viewer of the R Markdown document all of the code that you've written, but it also outputs all of all of the output that is the result of having run the code. So you will recall from earlier in the semester, I gave you some information in the form of our markdowns. Where are they? Well, I gave you the COVID analysis in our markdown. So let me just pull that up here really quickly. So I think I put this on Moodle, but just so we're clear, in the Kansas COVID analysis, can you guys see my PDF on your screen? Okay, so this is just a PDF file. And what I've done here at the beginning is it actually has everything that I did in the COVID masking case study that we did at the beginning of the semester dealing with masks. And it shows you, here's a code, here's some code that I did. And when I activated it, this is the output that R gave me. And then I ran this line of code, and this is the output that I gave me. I ran these lines of code, and this is what R gave me as output. And it gives you essentially what the code is, and then shows you what the output is. So the way I used to do assignments in this class before I myself learned about R Markdown is I would have each student turn in their data file, their script, and their write-up. And then what I would do to check whether or not you had done things correctly in R is I would upload your R script and your data file, and then I would basically just run your R script and see if your R script worked and gave the kinds of outputs that it should be doing. The advantage of doing this in R Markdown is that I do this, I can see what your code was. So in this case, there's a little graphing block that will then produce this figure. And so in an R Markdown document, in a single document, I can have not only what you wrote in terms of your code, but I can also see what the outputs of that code were. And so in one way, it just saves me a little bit of time because I'm not having to run as many scripts as there are in number of students in the class. And I don't have to keep track of each file, both the script file and the data file, because people will even structure their data files differently depending on who you are and how you're going about things. So in this situation, in this case, the document got really long because of some of the things that I was illustrating in the process of doing it, but it's not any big deal. I can quickly go through here and figure out, okay, here's another plotting block, another graphing block, and this graphing block produces this figure that we see down here. And so you'll turn in one of these. The other thing that R Markdown does, it allows you to make extensive comments that appear just as comments. These are what would normally be preceded by the pound sign in a regular script. So you'll turn in one of these, and then I can go through and see, well, what did you script and what is the output that it gave you when you did the scripting? And then there's also a write-up where you essentially describe what the data are like, describe what the analysis is like, and describe what the results of that analysis were. So in all of the cases, when we handle these write-ups, we're going to be behaving as if some researcher had come to us with a data set that they didn't know what to do with. They've asked us to analyze this. And our goal is to give them back an analysis that is correct in terms of all of its particulars. Is it done correctly? Did we check the assumptions that are necessary for a particular test to be run? Did we do any post hoc analyses to tease apart any differences that we do observe? A complete, full, correctly done analysis, but also written in such a way that the person who receives the analysis can make heads or tails of it. So written in language that is approachable to a person not trained in statistics, and also has summaries of the data included in that write-up such that it would aid that person in writing a paper if they were writing a paper for publication, or writing a report if this was a data set that had something to do with their job such that they needed to write a report describing what that data were like. And today what we're going to do is we're going to examine a peas analysis, an analysis having to do with pea growth, the growth of pea plants. And I've already analyzed the data, obviously, but I've also written up a summary of the data in the style of what I want you to be writing up for your first report. But it's written about the peas analysis rather than the analysis that you're actually going to do. And so that'll give you kind of a template of what I'm looking for in your reports. And one of the nice things about these reports is that they kind of take a standard form, as all kind of scientific reporting is about. So I'm not sure what your plans are after you graduate, but having taken a course that gives you a good grounding in statistics, chances are wherever you go after this, people will figure out that you know a little bit of something about doing statistics, and they will appear at your office at all kinds of times saying, hey, I've got this problem, can you help me out with this? And then you will be pulled into doing statistics for other people, either for pay in some cases, or for co-authorship on a paper if it's in a scientific setting where somebody's actually publishing a paper, or if it's one of your friends, just, you know, eternal gratitude for helping them out. But in all those cases, your ability to translate the results of your statistics to them in a way that they can digest it and use it is a skill in and of itself. And so that's part of what we'll be learning as we go through this, in addition to just learning how to do, for example, an analysis of variance. So do you guys have any questions about the path forward? All right, then, let's get started. There is a PowerPoint on the Moodle site under today's date, and there is also a small data set that we'll be working with. Have at least, if nothing else, have the data set handy, because we're going to be doing some things with that data set today in class. It's called Peas for Students, I think is the name of the data file, and it's stored as an Excel file. So today we begin doing statistics kind of in earnest. And we're going to be encountering increasingly more useful ways of doing statistics as opposed to something as simple as a t-test. Nothing wrong with t-tests, it's just that they have limitations and you can only analyze 2 samples. So today we're going to be talking about the analysis of variance. One of the things I want you to know is that the analysis of variance is not the analysis of means. Okay, so we're going to use an analysis of variance to detect differences among means, but we're not going to be analyzing the means themselves. We're going to be analyzing different forms of variance. This may not make much sense to you right now, but it's something that I want to put up in front of you right now, because when we get to a point in the analysis where we're talking about how things are actually calculated, it should become very apparent to you that it is an analysis of variances rather than an analysis of means. We're going to start doing a lot more in R. So before Friday, go back and review the basics of R. If you have to, go back and go through the R basic stuff that we did at the beginning of the semester, just to get yourself back in thinking about R. A couple of years ago, I ran across this quote by a blogger who blogs about doing scripting in R. And he's like, because I knew enough R to be clever, but not enough R to avoid being stupid, I created an entirely avoidable problem. It consumed a couple of hours of my wife's time. Of course, now she knows both about apply and sapply to functions in handling data in R. So you could argue that in the long run, I've probably still saved her time. I'd also say she's learned something about her husband's stubborn insistence on pretending he knows what he's doing, but she already is the world's leading expert on that topic. As you go through R, you're going to make mistakes. Everybody makes mistakes when they're scripting, when they're programming, et cetera, et cetera. And sometimes these mistakes will be really costly in terms of time, because you're taking one particular approach to a problem, and there's another approach that is much more efficient and actually doable, and you won't find that approach until you've actually spent some time looking for a solution. Now, remember that as you're scripting in R, there are all sorts of resources that are available. If you come by my office, either during office hours or by making an appointment, I have a book, the R book. It's a very thick book about R. Chances are you can find a lot of answers in this book to questions that you might have if you're struggling with something. There's also Stack Exchange on the internet. So if you go to Stack Exchange, you can find answers to R. There's a whole community online that deals with problems in R. Generally, those people are pretty helpful. Sometimes those people are really mean. But chances are, if you have a question about R, somebody else has already had that question about R, and there's already an answer to that question that exists on the internet. And then, of course, there's always me. You can come to me. I've done all of the analyses that I am expecting you to do, and so I should be able to help you figure out whatever it is that you're struggling with. The other thing is, when you get an error message, definitely pay attention to the error message. Read the error message. Try and figure out what the error message is trying to tell you. In some cases, our error messages are super helpful. And in some cases, our error messages are totally gibberish. And so sometimes it's really helpful to read the error message, and it will point you to exactly what the issue is. Other times it gives you a bunch of gobbledygook, and you're like, I have no idea what that error message means. But a lot of times paying attention to the error message will get you pretty far down the road. And then the last thing is, so many things break down in R simply because you made a simple typing mistake. So the first thing you should do when you get an error message is read the error message, and oftentimes it will point you to a section in your line of script where you simply misspelled something, mistyped something. As you know, some of us are better typists than the rest of us, and those of us who are not great typists have to go back and proofread our script pretty carefully. So, what does analysis of variance do? This is the P's data set. This is a experimenter who is growing pea plants under the influence of a plant hormone called an auxin. You don't need to know what an auxin is or what it does or anything else. All you really need to know is that one of the ways in which you might be able to, one of the ways in which auxins might alter the function of a plant is it might alter their ability to utilize sugar. And so in this case, we have a control group where the plants were grown without auxin in the soil and were just watered with straight water. Then we have a group where all the rest of these are grown with auxins in the soil. And then one group was given a 2% glucose solution, a 2% fructose solution, a 1% glucose. Why is that mistake still in this PowerPoint after all these years? I'm just going to fix it now and be done with it. a 2% glucose, a 2% fructose, a 1% glucose, 1% fructose, and a 2% sucrose. Now, you might ask yourself, well, why these treatments? Well, as it turns out, in chemistry, we have simple sugars and we can have compound sugars. So in this case, glucose is a sugar with a 6-membered ring, and fructose is also a sugar with a 6-membered ring. Sucrose, on the other hand, is a sugar where you have two simple sugars, bonded together to form a double sugar. And sucrose which is regular table sugar. Sucrose is a combination of glucose molecule bonded to fructose molecule. And so we have 2% solution of glucose by itself, a 2% solution of fructose by itself. We then have a 2% solution that is made up of half glucose and half fructose. And then we have a 2% solution of sucrose, which is glucose and fructose chemically bonded to one another. So we have 3 simple sugars, 1 compound sugar. We have 2 simple sugars that are pure in their mixture, 1 that is a combination of the 2, but then this one is a combination of the 2, but they're actually chemically bonded to one another. And so that's the reason for these different kinds of combinations of sugars. And if you were a statistician, one of your first questions to a person coming to you with these data should be, well, why are these the treatments? Like, what's your rationale for these treatments? Because I know a little bit about chemistry, I can figure out why these are the treatments. Plants need sugar in order to survive, as do we. And so then the question is, well, how are these plants utilizing these kinds of sugars, and what does auxins have to do with that? And then the numbers here are simply how much the plant grew over some period of time. And if I recall correctly, these are in millimeters, but it doesn't really matter. But this is all just what the researcher refers to as growth increment. Over the same period of time, under the same conditions, how much did each of these plants grow? You guys, are you clear on kind of what this researcher is doing at this point? Yes, no. Yeah, okay, all right. So one of the questions that you might ask is, you could say, well, Dr. Kowalski, you just took us through doing t-tests. Well, we could actually just do a set of t-tests. We could compare the control to the 2% glucose, then we could compare control to 2% fructose, then we could compare control to 1% glucose, 1% fructose. We could compare control to 2% sucrose, compare glucose to fructose, glucose to the 2, glucose to sucrose, compare fructose to glucose and fructose, fructose to sucrose, the mixture to sucrose, et cetera, et cetera. So one of the questions that you might ask yourself is, why not just do multiple t-tests rather than learn a new technique? We already know how to do a t-test. What would your answer be?
Wouldn't it be about the averages instead of the variance? If we did a t-test instead of a variance test?
Well, yeah, but that's just assuming that you would want to do an analysis of variance. Why even learn the analysis of variance if we can analyze these data with just a bunch of t-tests?
To independently compare them? I don't know. That's a good point.
When you do a t-test, you calculate the t-statistic, and then you compare that t-statistic to a table t-value. And when you look up that t-value in a table, you basically look at the combined degrees of freedom, and you also look at the alpha level. What is alpha level telling you again? Somebody tell me what alpha is.
It's the error rate, right?
That's the type 1 error rate. It's the probability of saying that two means are different when in actuality they are the same. So it's essentially saying this. And we only want to make that mistake about 5% of the time. That's typically sort of the industry standard at which you test. So the problem with doing multiple t-tests is that the more comparisons you make, the more likely you are to make a type 1 error. And here's the reason why. Type 1 error rate is additive. So if we go back and say, this is A, B, C, D, and E, there's a type 1 error rate when we test to see if A is different from B. Then there's a type 1 error rate when we test to see if A is different from C, A is different from D, and A is different from E. Then there's a type 1 error rate having to do with B versus C. There's another type 1 error rate having to do with B versus D, and then B versus E. And so when you get to the end of all of this, the probability of a union of events is the sum of their individual probabilities. So this comes from basic probability theory. So if you do multiple t-tests, you're asking what is the probability of a type 1 error in the first comparison, A versus B, then A versus C, A versus D, A versus E, then B versus C, B versus D, B versus E, B, C versus D, C versus E, and then B versus E. So that's 10 comparisons. Each of these comparisons have a 0.05 type 1 error rate. And so if we take 0.05 and multiply it times 10, our type 1 error rate for all of those combined comparisons is now 0.5. So across the entire experiment, we have a 50% chance of making a type 1 error. Well, if that's the case, what's a more efficient way of figuring out if these means are different from one another? Way more efficient in terms of time. If you want to know if the mean of A is different from the mean of B, rather than do all the statistics, what could you just do given that you have a 50% type 1 error rate? What's something that has a standard, 50% chance? Chance?
Coin flip?
Yeah, you might as well say, well, is A different from B? Flip a coin. If it's heads, they're different. If it's tails, they're not. A versus C, flip a coin. If it's heads, there's a difference, and if tails, there's not. That's the power that you get from doing a lot of different t-tests. You reduce your ability to actually see the difference because your type 1 error rate is really high. And so what you need is you need a method by which you can test all these comparisons while maintaining your experiment-wide type 1 error rate at 0.05. And that's what analysis of variance does. Analysis of variance was invented by a guy named Ronald A. Fisher, and he was actually interested in the phenotypes among the offspring of different kinds of crosses. He was a geneticist, and so he was interested in genetic variation, and he invented analysis of variance as a method for analyzing the outcomes of genetic crosses that he was doing. So in an analysis of variance, we can think of null and alternative hypotheses, just like we thought of them in a t-test. And the null hypothesis is that all populations share the same mean, and any differences between sample means are caused just by random sampling error, versus the alternative hypothesis, which is that all populations do not share the same mean. At least one population has a mean different from the rest. So we're making hypotheses about the means but we're going to determine whether those means are different by making analyses of the variation around the means. So the null is that the means are all the same. The alternative is that at least two of the means are different. No telling which two are different, but it will detect whether there are any differences. So another way of thinking about the hypothesis is by basically saying that all the sample means were drawn from the same underlying statistical population, getting back to this idea of what a statistical population is, versus the alternative that at least one sample mean was drawn from an underlying statistical population that is different from at least one of the remaining populations. And so it doesn't really matter how you think about these hypotheses. Both of these are valid ways of thinking about the null and the alternative hypothesis. So one way to think about analysis of variance is a comparison of different models. And in this case, we can think about different kinds of comparisons. These comparisons are comparisons done in the sums of the squared deviations from something of each observation from the grand mean, each observation from that observation sample mean, and then the remainder is whatever is due to the alternative hypothesis. So this is the total sums of squares, the within sample sums of squares, and then whatever is left over is the among sample sums of squares. And this is actually what we're interested in, is how big are the differences caused due to the different treatments. That's what the among sample sums of squares are. So this probably doesn't make a ton of sense. A different way of thinking about this is let's just make a plot of these data. And so what I've done is on the x-axis here is growth increment, and on the y-axis are the different treatments. And so for each of these treatments, we could basically plot where the data fall. So for the control, we have one plant that grew 65 millimeters. We have 3 plants that grew 67, one plant that grew 68, one plant that grew 70, one plant that grew 71, one plant, or 2 plants that grew 75, and one plant that grew 76. So this is one way to visualize the data. We could look at the visualization for 2% glucose. We have 2 that grew 57, 1 that grew 58, 2 that grew 59, 3 that grew 60, 1 that grew 61, and 1 that grew 62. We can look at the same for the rest of these as well. The 2% fructose, 1% glucose, 1% fructose, and 2% sucrose. What are some things that jump out at you here at the beginning just by looking at these data?
That the control group had the greatest growth out of any of the treatments.
Okay, so it looks like the control group might have higher growth than any of the other groups. What other things jump out at you? Somebody other than Hayden? Logan.
Blue, red, and green all had like the same relative growth.
Yeah, it looks like the 1% glucose, 1% fructose, the 2% fructose, and 2% glucose. Those all look pretty similar to one another, don't they, in terms of their overall position relative to the x-axis? What else is similar among those three, Jonathan?
Well, I mean, if you look at the 2% glucose, 2% fructose, then you can go 1 and 1%. I mean, they're kind of the same in what you've added. I mean,
okay, so they're kind of so I'm thinking about their distributions at this point.
Oh, okay. Well, I mean, other than them being fairly the same, I mean, I don't know what else you could add to that.
So it looks like their position is relatively the same. What's their variation like?
Well, if you look at their max and min, they're pretty similar. But if you were to look at like the mode on them and stuff, they drop a little bit. I mean, you got 60 for the 2% glucose. Looks like you got 70, or looks like you got 58 fructose,
Yeah.
and then
So their modes are slightly different, but their overall range of variation is very similar. Haley, is this range of variation similar to sucrose and similar to the control?
I would say it's more similar to control, or yeah, I would say it's more similar to control because instead of having like left skew data, it's more like in the middle, like control.
But look at total variation though. This spans from like 56 to 62. This spans from 63 to 68. So that's a range of 5. That's a range of 6. This is a range of 6. This is a range of 6. Look at the range on the control. Is the control more variable, less variable, or about the same as the other ones?
It's more than the other ones.
Yeah, it's more variable. Okay, so you notice two things are going on in these data. One is that the groups themselves, the treatments themselves, seem to be different from one another in some cases, but you'll also notice that some samples have less variance than other samples within this particular experiment. Now, one of the things that we could do is we could calculate the average of each of these treatments. And when you look at the averages of each of these treatments, this is where they fall. And I've also plotted 95% confidence intervals to either side of these. Now, when you plot the 95% confidence interval of the mean, somebody remind me again what the 95% confidence interval of the mean is if you were trying to explain this to your grandmother or your mother. Samuel, take a shot at it. And you can explain it within the context of the control, for example. So this is the mean and the 95% confidence interval of the mean. What does that mean? What does a 95% confidence interval indicate, given that this is the 95% confidence interval for the growth increment of a bunch of peas grown that are just simply watered with regular water?
Doesn't it show like, I don't know how to put it in words. Doesn't it basically show like the range of numbers that were like pretty sure it'll fall under like 95% of the time?
What will be within that range 95% of the time?
Like the mean, correct?
Yeah. So if we took 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, if we took 10 pea plants and planted them in the ground again, 10 different pea plants, planted them in the ground again, watered them the same way, gave them the same amount of light, et cetera, we would calculate a mean, and there's a 95% chance that that mean would fall within this range. There's a 5% chance that that mean would fall outside this range. If we took 1,000 plants, planted them in 100 plots of 10 plants each, calculated the mean growth for each of those plots, we would find that 95% of those plots would have average growth increments that fell within this range, and 5% of them would fall outside of that range. So yeah, that's exactly what the 95% confidence interval is about. And that explains why there are observations in this particular sample that fall outside that range. This is a confidence interval for the distribution of sample means, not the distribution of the actual data. So remember that when we think about what these 95% confidence intervals are. Now that you know that these are the 95% confidence intervals, knowing what a 95% confidence interval is, what would you predict the differences among the means are? Anybody? Hannah?
Can you repeat the question, please?
Now that we know what the 95% confidence interval of the mean is for each of these different treatments, can you make some educated guesses as to which means are different from one another and which means are not different from one another?
Yeah, the control is going to be different than the other 3 or 4.
Mm-hmm. What else?
And is the 1% glucose plus 1% fructose added different?
That's the blue one, so probably not. If we run the means up, we see that this mean is right at the edge of this mean's confidence interval, but what other mean looks different from other means? So you said that the control is different from everybody else. What else is different?
The 2%.
Yes, 2% sucrose is different from who?
The pretty much the blue, red, and green.
Mm-hmm.
And the control.
And the control, yeah. So just based on simple t-test sorts of ways of thinking about this, does this mean fall within the confidence intervals of any of the other means? It doesn't, so you would say that that one's different. Does this mean fall within the confidence interval of any of the others? You would say, no, it doesn't. The green mean falls outside the confidence interval of the blue mean, but the blue mean falls within the confidence interval of the green mean, or very close to it. These are all estimates because I was eyeballing this. This would be more precise if I had done this in R, but unfortunately, the goal of this figure is slightly different. So it looks like red, blue, and green are not different from one another, but they are different from yellow, and those are all different from black. So what an analysis of variance is going to do is it's going to evaluate all of these potential differences at the same time, simultaneously. And the way that it's going to do this is by looking at variances. So the null hypothesis is that all of these samples came from the same underlying statistical population. If that were the case, there's just one big sample, and that big sample has a grand mean. So the X with the double bars over it is the grand mean. It is the mean of all of the data combined across all treatments. And when we calculate the grand mean, it's right down here around 62. And so that's what it looks like. This is where the grand mean is. And the grand mean is useful because this would be the average that we would expect if all of the treatments were drawn from the same underlying statistical population. And we're testing to see if that's the case, or is the control drawn from one population and everybody else is drawn from a different population? Is 2% sucrose drawn from one population and control and these three are drawn from a different population? Is the blue sample drawn from a blue population and the red sample drawn from a different population? And so we can think of the grand mean as the average we should expect to see if there are no differences among these different treatments. So now we want to know, to what degree do each of these sample means differ from the grand mean? And so one of the things that we can do is we can actually calculate how does each sample mean differ from the grand mean, square that, multiply each of those times the the sample size for that, and sum those up. And that gives us the sums of squares among the different treatments. So we can ask, to what degree is the control mean different from the grand mean? To what degree is the 2% glucose mean different from the grand mean? Red mean different from grand mean? Blue mean different from grand mean? Yellow mean different from grand mean? That then tells us, how do each of these treatments differ from the grand mean? That is the sums of squares among. And that's this portion of this complicated table here. But we can also ask ourselves, how much variation is within each sample? So we can ask, to what degree is this observation different from its sample mean? To what degree are these three observations different from its sample mean? To what degree is this data point different from its mean? How is this one different from the mean? How is this one different from the mean? How are these two different from the mean? How is this one different from the mean? That's within sample variation. And that's simply the same way we calculated within sample variation when we were just dealing with a single sample. The sum of the individual values minus the mean squared. This is the sum square within. And we would do that for this data set. We would do that for this data set, this treatment, and this treatment. Are these two different from the green mean? This one different from the green mean? These two different from the green mean? These three different from the green mean? This one different from the green mean? This one different from the green mean? That is the sums of squares within. It's the individual values within a treatment relative to the treatment mean squared, added up across all the values within a treatment and added up across all the treatments. That's why there's two summations. This first summation on the right-hand side is summing within a treatment, and then this is summing across treatments. So we have at the top of this figure, we have 4 different treatments, and so this would be summed across 1 to 4. This is summed across 1 to however many samples are within a given sample, like within a given treatment. So you're summing within this treatment, then you're adding that to the sum within this treatment, you're adding that to this treatment, to that treatment, and to that treatment. And so you get a within sample variance estimate that has the within sample variation for all 5 of your different samples. Now, if you add together the sum squares among and the sum squares within, that should equal the sum squares total. Because the variation, the overall variation in this data set, if we look at all of the variation in the data set, it's influenced by 2 forms of variation. It's influenced by how the treatment drags the within sample observations away from the grand mean. How does 2% glucose drag these observations away from the grand mean? How does 2% fructose grab these observations away from the grand mean? How does 2% sucrose drag these observations away from the grand mean? So that's one form of variation. That's the among sample variation. But then there's also variation that is within the 2% sucrose, within the 1% glucose, 1% fructose, within the 2% fructose, within the 2% glucose, and within the 2%, within the control. That's another form of variation. And that's all the variation that there is to be had. And that's what the sums of squares total is. Now, you can calculate sums of squares total by calculating sum squares among and sum of squares within and adding those two things together. But you can also calculate the sums of squares total by asking yourself, to what degree does this observation in the control differ from the grand mean? To what degree do these three values from the control differ from the grand mean? How does this value differ from the grand mean? This value different from the grand mean? This value differ from the grand mean? This value differ from the grand mean? This value differ from the grand mean? This value different from the grand mean? Each of those deviations from the grand mean, you can square them. You can then ask how green differs from the grand mean, how this one from the green differs from the grand mean. These three differ from the grand mean. These two differ from the grand mean. This one differs from the grand mean. And these two differ from the grand mean. All of those deviations squared, and do that for all of the data points. That's another way of calculating sums of squares total, is by essentially comparing how each individual value differs from the grand mean, squaring that value, and adding those up across everything. When you do those two things, it should equal the same as sums of squares total if you just take some squares among and some squares within and add them together. So once again, one of the things I like about statistics is there are all these internal checks that you can apply as you're going through this. So keep in mind that when you do the sums of squares among, you have to weight the sums of squares times the sample size. It doesn't make that big of a difference in this case because each of these samples has 10 observations. But as soon as you get different sample sizes in your different samples, that will make a difference. So then the way you calculate an analysis of variance is you just get the sums of squares among, the sums of squares within. You divide those by the degrees of freedom to get the mean squared among and mean squared within. And this mean squared among and mean squared within is just the same as this is a variance estimate. It's the sums of squares divided by the degrees of freedom. The sums of squares divided by the degrees of freedom. And then you assess this basically using the F statistic, which is looking at how big is the variation among the means compared to the variation within the means. And this is what you actually test. And you can look up F statistics in a table where you evaluate it at a particular alpha level, and you evaluate it with the numerator degrees of freedom and the denominator degrees of freedom. And that will then give you an F statistic. And if your observed F statistic is bigger than the one that's in the table, then at least two of those means are different. And once again, the sum of squares total is just the combination of these two, although you know now that you can also calculate that independently. What happened there? That's weird. Okay. So what I want you to do now is go to the Excel file that I put on the Moodle side. So it's on a different screen. Yeah. All right, can you guys see my Excel spreadsheet? You guys have your Excel spreadsheets up on your computers? Okay, so here's the control. Here's 2% glucose, 2% fructose, 1% glucose fructose, 2% sucrose. What's the first thing that we need to know for each of these things?
The average, the mean.
Yep, the average. So we can calculate the average by just asking Excel for the average for that range of cells. Once we have that, we can just copy that formula over to each of these other cells at the bottom. And that gives you the averages. Now, what do we need next? We need the sum of squares among, and we need the sum of the squares within. So which one do you want to do first?
Among.
Okay, so if we're going to do among, so let's put ourselves a little marker over here somewhere. Sum squares among. What do we need in order to calculate sums of squares? Actually, let's move this down a couple. What do we need to know in order to do sums of squares among? Well, we need to know the mean of each sample. We need to know the grand mean, and we need to know the sample size, right? So go back to our Excel file. We can get the sample size by simply doing count, and we can count the number of observations in this sample. We can do that for all the rest, although in this case it's easy because they're 10. But still, but what else do we need? So we know the sample means because these are the sample means. We know the sample sizes. What's the other thing that we need to know?
We need to get the mean of all the means.
Yeah, we need to get the grand mean of the entire data set. And don't just think that you can add up the means and divide by 4. In this case, you can. So we could actually do this. We could basically say the grand mean is equal to the average of this, 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 and this. And that would be the grand mean, which is also the same as taking the average of this column plus this column plus this column. I'm using the command key to group these all separately. And this column. So in this case, they turn out to be the same, 61.94. I remember I told you it was close to 62. But the only reason that those two operations give you the same value is that these are all the same sample size. If they were different sample sizes, it wouldn't give you the same answer. So I'm going to do this, 76 and 68. I'm going to put these two things over here so that I can remember that there was a 76 here and a 68 here. So that's changed the mean of the control, but it's also reduced the sample size of the control. And now this is still just the mean of the means. And then this is now the mean of all of those values that we just looked at. But now the means are different because this one has fewer observations than the rest. As a result, it has less influence on the overall grand mean. And that's what leads to these two things having different values. So don't, when you work out your homework assignment on this, don't just average the means. You need to actually average all of the data that is in the analysis in order to get the right grand mean. If you simply average the means, chances are it's going to give you the wrong answer. So we're going to get rid of that one that averages the means, because that is the wrong way of going about doing this. So now, how do we calculate the sums of squares among? Well, it's simply the sample size multiplied times the sample mean minus the grand mean squared. Whoa, what happened there? Something is wrong. Yeah, I needed a multiplication thing in there. So in this case, this would also then be the sample size times the sample mean minus the grand mean squared. We would do a similar thing here, equals sample size times the sample mean minus the grand mean squared. And we can simplify this by just copying over. Copying over. And we have to change this because this is not O17. This is actually N17. And this is not Q17. This is, once again, N17. And then what we do is we simply add those things together. And that's the sum of squares among, 1077.32. So the sum of squares among is the weighted sum of the squared deviations of the individual sample means from the grand mean. And that is exactly what this formula says. It's the weighted squared deviations of the sample means from the grand mean summed up across all the samples. That is what sums of squares among is. So when we have sums of squares among, what else do we need to have?
Sum of squares within.
Sum of squares within. We already know how to do that, right? What's the sum of squares within? It's the mean, or the value minus the mean squared, right? So we can actually, instead of putting A14 in there, we can put 70.1. And then as a result of that, we can just copy those down. We can copy that across and just basically change what the mean value is to 59.3. And then copy it down. Do the same thing over again.
So that's
And then-
the sample minus the grand mean squared, correct?
Not the grand mean, the sample mean, because we're doing within sample variation, which is why I'm having to change each of these to the sample mean. So the sample mean in this case is 58.2. Over here for the 1% glucose, 1% fructose, the sample mean is 58. And over here, the sample mean is 64.1. Once I have all of those done, I can then just copy them down. So this is within sample variation. So you see this one is 0 because 58 is the same as the sample mean. I can copy those down. I can copy those down. And then I want to sum them. Within the sample. And I want to sum them for each of these. And then I want to sum across that, across all of the treatments. And that should give me some squares within. So now if I want sum of squares total, all I have to do is add sum of squares within plus sum of squares among, and that will give me sum squares total.
Can you go back and click on just one of the boxes for the formula again?
Which one? These?
5.2, yeah.
Okay. Yep. It's simply the value minus the sample mean squared. It's the sum of squares. It's simply the sum of the squared deviations of the actual value from the mean, the sample mean in this case, not the grand mean, but the sample mean. So it's the numerator of your regular variance estimate that you've already calculated multiple times now. And then once you've done that within samples, you add up the stuff within each sample, and then you add across samples to get the total sum of squares within. Who was that asking the question? Is that Hayden?
Yeah, I was. I didn't
Okay, all right.
I did go back and put in the dollar signs.
Okay, we're out of time. So what I want you to do in preparation for Friday is I want you to calculate the sums of squares total independently of just adding sum squares within, sum squares among, and verify that the sum squares total estimate that you get is the same for calculating it independently. Remember, the sum squares total will be the sum of the squares, the sum of the square deviation of each individual observation minus the grand mean summed across all treatments. And I find this easiest to do in a little side matrix where I do that in a matrix that is as big as my data file is. But you can do it whatever way you do. You're just going to basically take 75 minus the grand mean squared plus 67 minus the grand mean squared plus 70 minus the grand mean squared through all of these until you get down to 67 minus the grand mean squared. And that would be sum of squares total, and it should equal 1322.82. And so do that in preparation for Friday, and we'll start there on Friday. in class in White Science 205. Questions? Are you following the way we're going about doing this okay? Okay. All right. Sum of squares total on Friday.
Did you just say when we're calculating that it'll be the like for the control group it'd be 75 minus the grand mean squared, and then you do that all and then add them all up?
Yep. Yeah. So it's the individual values minus the grand mean squared, added up across the entire data set. Yep.
Perfect.
All right.
So within when we're calculating the standard sum of squares within, is that just the sum of the second column that we just filled out, which would be the mean of the sample, so observed minus the mean squared?
Yeah. And so you do that within each sample, but then you, in this case, you have 50 of those and you add all of those up.
Add all those up. Okay. See that I'll have to show it to you on Friday. As my sum of squares within, I'm getting 227.5 instead of 249, like I believe is what you have.
I have 245.5.
Yeah, and I can come see you today, or I can come and we can just maybe just. I obviously just made like a little slight error. I can check.
Yeah, check for a slight error. When do you want to come by today?
Can I come by before 11:15?
Yeah, why don't you just come during that Joule time slot?
Perfect.
All right. See you then.
Thank you very much.
Sure.